Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at understanding how errors in DNA replication can give rise to mutations. We'll also look at how cystic fibrosis results from one of a number of possible gene mutations. We'll also understand how the expression of a gene mutation in people with cystic fibrosis impairs the functioning of the gas exchange, digestive, and reproductive system. So a gene mutation is an unexpected change in the sequence of DNA that could result in a different polypeptide being produced. These errors can take place mostly during DNA replication. As protein structures are determined by DNA sequences, mutations in the DNA sequence can result in alteration in the structure of the polypeptide produced. However, most mutations do not cause too much change in the structure, and this is because the genetic code is degenerate. The different ways mutation can take place in DNA sequence are insertion, deletion, substitution, duplication, and inversion. Insertion of nucleotides. So insertion is when the mutation that occurs when a nucleotide is randomly inserted into the DNA sequence. This results in a change in the base triplets, resulting in a change in the amino acid produced. This therefore affects the polypeptide produced. Insertion changes the triplets further on the DNA sequence, and this is known as a frameshift mutation, as it changes the codon where the mutation has occurred and all the codons further down the sequence. Deletion of nucleotides. So deletion is when a nucleotide is randomly deleted from the DNA sequence. This also changes the triplet where deletion has taken place and the codons further down the DNA sequence. This is also known as an example of frameshift mutation. This also affects the amino acids produced and therefore the polypeptide. Looking at substitution of nucleotides. So substitution is when a base in the DNA sequence is swapped for a different base. This only changes the codon where the mutation has taken place, and the codons further down the DNA sequence are not affected. Substitution can be of three types. They can be silent, missense, and nonsense. Silent is when the altered nucleotide does not change the amino acid produced due to its degenerate nature. So degenerate, if you guys remember, is when different combinations of nucleotides can result in the same amino acid. So that means there's no change in your actual nucleotide sequence. So the polypeptide produced is not affected. Missense is when a single amino acid is altered. And nonsense is when the mutation creates a premature stop codon, hence the complete polypeptide chain is not produced. So duplication is when more than one copy of a gene or a region of a chromosome is produced. As the original version of the gene remains intact, the mutation is not harmful. However, the second copy can develop into mutations which can cause harm. Alternatively, it is also possible that it can lead to advantageous traits which can be beneficial for evolution. This type of mutation is more common in plants than it is in other organisms. Inversion. So inversion mutations take place during meiosis. The DNA of a single gene is cut into two places and it is inverted 180 degrees and rejoined to the same position in the gene. This results in the DNA sequence being read the other way around, hence affecting the amino acid sequence produced and hence the polypeptide as a result. Since the original gene is affected, it can no longer be expressed from that chromosome, possibly causing harm. So now we're looking at cystic fibrosis, which is a genetic condition that you need to learn about in topic 2. So genetic disorders are usually caused by mutations in a gene that alters the protein produced and therefore the phenotype. So just to remind you guys, phenotype is the physical characteristic as a result of a specific gene. So cystic fibrosis is a genetic condition caused by recessive alleles of the CFTR gene on chromosome 7. This gene codes for the production of chloride ion channels required for secretion of sweat, mucus, and digestive juices. A mutation in the gene leads to the production of non-functional chloride channels, which reduce the movement of water by osmosis. This results in the body producing huge amounts of thick, sticky mucus in the air passage, digestive tract, and reproductive system. Mutations in the CFTR gene that can lead to cystic fibrosis. In fact, there are 1,000 different mutations that can lead to cystic fibrosis. As cystic fibrosis is a recessive condition, which means that both the alleles present should be recessive, people who are heterozygous will be carriers, and people who are homozygous will have the disorder. And you need to be homozygous recessive to have the disorder. And just to remind you guys, heterozygous is when you have one dominant allele and one recessive allele. So if two carriers have a child, there is a 25% chance the baby will have the disorder. And if one of the parents is the carrier, there is no chance to develop cystic fibrosis. And you guys can check this out yourselves by drawing a Punnett square, where in case number 1, I would actually recommend you guys to do this and check, by the way. So case number 1, where both parents are carriers, and then you need to look at case number 2, where only one of the parents is a carrier, and you can look at the chance of the baby having cystic fibrosis. The respiratory system and how it's affected by cystic fibrosis. So mucus in the lungs prevents infection by trapping microorganisms. This mucus is wafted up the windpipe through the cilia. People affected by cystic fibrosis have thick, sticky mucus, hence the cilia is unable to waft up the mucus. This results in microorganisms not being effectively removed, resulting in an increased risk of infections in the lungs. The thick mucus can also limit gas exchange as the airways are blocked, resulting in breathing difficulties as the surface area for gas exchange is reduced. Physiotherapy is often offered to lose the mucus in the airways and improve gas exchange. How cystic fibrosis affects the digestive system. So thick mucus blocks the tube to the pancreas, preventing digestive enzymes from entering the small intestine. This reduces the efficiency of digestion. The mucus can also cause cysts to grow in the pancreas, which inhibits the production of enzymes further. The intestine linings are also coated in thick mucus, further minimizing absorption. So if you guys know from GCSE, the small intestine has a large surface area which is really important for absorbing the nutrients back into the bloodstream. But because this is covered in thick mucus, this ability to absorb is massively reduced. How the reproductive system is affected. So mucus is secreted to prevent infection and regulate the progress of sperm through the reproductive tract after intercourse. The thick mucus in people affected by cystic fibrosis results in men the tubes being blocked by the sperm from passing through the reproductive tract, and in women the thickened cervical mucus prevents the sperm from reaching the oviduct, hence preventing the egg from being fertilized. So that is all from Professor Snap today. I think this is quite a detail-rich video, so you might want to watch it twice or more, so feel free to do that. If you have any questions, let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share and subscribe.